everybody. I'm Eleonora Cariolato, and I'm going to present my thesis work on mitochondrial DNA genetic diversity and phylogeography of Laternula elliptica in the Western Antarctic Peninsula. My promoter is Francesca Pasotti, and my co-promoter is this project is part of a bigger project, the Rexo project. So as you might know, the Antarctic ecosystem is characterized by a very extreme environment, very cold with high seasonality, and also it's characterized by a long isolation and a particularly evolutionary history that overall led to a local adaptation of its organisms and also a high level of endemism and a high biodiversity. However, current climate changes and anthropogenic pressure are threatening this ecosystem. For example, changes in the Southern Ocean current system that has been detected in the past decades. Its organisms are facing difficulties in coping with the increase of temperature and the decrease of pH. And there is an increase of the development of the new habitats due to the melting of sea ice and the retreat of ice sheets. And also an increase of the establishment of invasive species and a shift in the species distribution due to the increase of tourism, especially in the Antarctic Peninsula. Therefore, in this light, studies that focus on the phylogeography and the population connectivity are very important to protect and to mitigate the effect of climate changes in the endemic species. So in this context, my work focused on Laternula elliptica, which is an endemic bivalve from the Southern Ocean that is a broadcasting free spawning with lecitotrophic larvae. It's a shallow water bivalve, and it's also a filter feeder, so it's considered as a keystone organisms. This study also employed molecular tools, such as the mitochondrial DNA as a genetic marker, and next generation sequences techniques that allows to have a low-cost and high-throughput methods that is especially indicated for non-model organisms such as Laternula elliptica. Three main objectives were drawn for this master thesis. The first one is investigating the population structure of Laternula elliptica in the Western Antarctic Peninsula. The second one was investigated the phylogeography of the target species in the same region. And the third one was trying to have an insight on the Glacial Refugia by looking at the molecular signature, where a Refugia is an in situ survival hotspot. So, as I said previously, the sampling sites are focused part of the Antarctic Peninsula. There are 6 different populations. And also, it was supposed to include an extra population from outside the peninsula. However, due to COVID pandemic, it was not possible to obtain the sequences in time. The mitochondrial DNA extraction protocol started with the tissue preparation from a somatic tissue, like the siphon, the mitochondrial DNA extraction, the quality control of the extraction, the DNA concentration measurement, a long-range PCR amplification that employed 4 different primer pairs, in order to amplify 4 different fragments and to cover the whole mitochondrial genome, quality control of the PCR products, and the good quality products were sent to be sequenced with Illumia next generation sequencer. Once obtained back the sequences, they were processed using Genius Prime, DNA SP, and JModel test softwares to assembly, map the reference, and run nucleotide substitution models. MEGA7 was employed to calculate genetic distances of both within and between population. PopArt software was employed to build haplotype networks. And finally, RStudio was employed to build a graphic representation of the genetic distances resulting from MEGA7. Moving to the results, I'm showing you the successful sequences rate and the mitochondrial regions. So the average length of the sequences is around 3,500 base pair, and a consensus threshold of 75% was the one selected and extracted from Genius Prime. However, there was not an equal number of sequences across the population and the mitochondrial regions. For example, as you can see, mitochondrial region 26 and region 4 has a lower PCR amplification and sequence assembly success, and therefore the number of population and number of sequences that include these two regions were fewer compared to the other ones. Moreover, region 1 and region 4 include sequences that were previously processed before the start of my thesis. Okay, so looking now at the estimation of the genetic distances, we're looking at the within population first. So in this graph, on the y-axis, we have the AGD, average genetic distance in percentage, and on the x-axis, we have the different population, where each color represents a different mitochondrial region. So first of all, as you can see, the AGD value do not exceed 0.4%, and we have the Deception Island population and Kindred Island population show the highest AGD value, while Rotterdam Station has the lowest one. The difference, for example, between Deception Island and Rotterdam is around 0.14%. Looking now at the between population, we have a similar graph with the same y-axis, and on the x-axis, we have the different population pairs. And again, the AGD percentage do not exceed the 0.3%. As you can see, the difference between the Green Reef and Deception Island population has the highest AGD percentage, while Melchior Island and Rotterdam Station has the lowest one. And the majority of the population pair have value that is in the range of 0.14% and 0.19%. As you have probably seen the previous graph, the AGD percentage value varies also according to the mitochondrial region investigated. So in order to see the molecular pattern of these differences, and due to the fact that there was not an equal sample size across the population and the mitochondrial region, subsets of equal number of sequences were created in order to compare the effect of the sample size on the AGD value. So again, looking first of all at the within population, on the left graph, we have a comparison of mitochondrial region 26 and 1 has the same sample size, and with two populations. And on the right graph, we have a comparison between region 4 and region 1 with one population. In both scenarios, we have the region 26 and region 4 show a slightly higher AGD percentage value compared to region 1. The same was done for between populations, and we have, again, on the left graph, a comparison between the region 26 and the region 1 and the region 2 for two population pairs. And on the right graph, we have region 4 compared with region 1 for one population pair. Similarly to the previous slide, we see that mitochondrial region 26 and 4 show a slightly higher AGD percentage compared to region 1 and 2 when they have the same sample size. Finally, I'm going to show you the haplotype network that were built. I'm going to show you the mitochondrial region 1 and median join because it's the one that has the bigger data set, so the most informative. And so each circle represents a single haplotype, and the size of the circle indicates the number of samples that share the same haplotype. The different color indicates the different populations, and the small horizontal black lines indicate the mutational step. So in this graph, in this network, we can see that two dominant haplotypes can be identified, Rotterdam 53 and Green Reef 03. And the majority of the Rotterdam sample are share the same haplotype or have few mutational steps. Moreover, two sequences that belong to the same sample, the Deception Island 03, and they were processed in two different times. They were kept separated during the analysis to see where they were located in the haplotype network. And surprisingly, they're far apart from each other. Overall, no obvious geographic pattern can be observed in this network. So, wrapping up all the results, I would like to start my discussion by evaluating the molecular methods employed. So, according to our results, it seems that COX-1, 16S, and cytochrome B genes are slightly more informative, and they are the genes that were covered by mitochondrial region 26 and 4. And actually, these genes are the ones that are widely employed for this type of study. The two Deception Island 03 sequence that were kept separated actually show very low AGD percentage. Even if this is not significant per se, however, these values falls in the range of the AGD of this study. Therefore, the position far apart in the network is probably due to an artifact. Moreover, the low success rate of the PCR can be due for many different technical reasons, like the quality of the sample, the genetic variation of primary binding sites, and also the possibility of pseudogenes in COX-1 of Laternula elliptica. And finally, also the very low genetic distance value, they're very low compared to similar studies, can also be due to technical reasons, like not having employed the most suitable genetic marker, and also to the fact that probably the mitochondrial DNA of Laternula elliptica is not really variable. Nevertheless, we can still discuss the phylogeography and the connectivity of Laternula elliptica. So our results overall assess that there's no difference in the AGD values of within and between population calculation, that there is a very low AGD value in the region, and also that no geographic pattern or no obvious geographic pattern was observed. And this actually can be supported by the reproductive system of Laternula elliptica, because it's a broadcasting spawning species, and also has a licitotrophic larvae that can stay viable in the water column for up to 18 months, so it has a great dispersive capacity. And also the geographic area investigated is pretty limited. Finally, looking at the molecular signature of the Glacial Refugia, our haplotype network actually reassembled two scenarios. So the first one, the top one, is a continental shelf refugia, and the bottom one is a deep sea refugia. Both of them are for species with dispersal stage like Laternula elliptica. And so since this network sits in between these two scenarios, three hypotheses were formulated. The first one is a deep sea refugia that consists in refugia located in the deep sea that recolonize the continental shelf. However, Laternula elliptica is a shallow water, and also some kind of genetic connectivity signature should be inferred when looking at the molecular analysis. Therefore, further study in order to test this hypothesis should include a greater depth ranges. The second hypothesis is a multiple continental shelf refugia that consists in multiple small in situ refugia in the continental shelf that has a secondary contact. Again, to test this hypothesis, further studies should include population from other Antarctic regions in order to see how this pattern can change with greater distances. Finally, the third hypothesis is island refugia that consists in a refugia located in a peri-Antarctic island that through larvae dispersal stage can recolonize the Antarctic Peninsula, for example, and its islands. Again, in order to support this hypothesis, further studies should include population from the peri-Antarctic islands, such as the South Orkney Island. In conclusion, this master thesis work assessed low genetic diversity for Antarctic species, Laternula elliptica, in the West Antarctic Peninsula, suggesting a good gene flow and good connectivity in the region. No obvious geographic pattern was observed in the region, and also looking at the haplotype network, three refugia hypotheses were formulated for the molecular signature. However, since only one Antarctic region was investigated and there was a limitation with the sample size and that different technical difficulties were encountered, we suggest a further study in order to support these preliminary results should include more suitable genetic markers and equal sample sizes and also more Antarctic regions. Thank you very much, and yes.